Welcome to Planetterrian Daily, episode 113. Today is July 7th, 2026. Some interesting findings to cover today. A prospective study of 73 adults links subtle heart changes to gray matter damage and memory issues even without heart failure. A prospective study of 73 adults found cardiac function tied to gray matter alterations and memory performance even without heart failure. The work highlights ongoing heart-brain interactions across normal aging rather than only disease states. Researchers note the need for larger replication cohorts to confirm the patterns. The findings add to tracked questions on how brain aging processes intersect with other organ systems. Analysis of some of the planet's oldest volcanic rocks shows water played a significant role in driving volcanic activity and reshaping the planetary interior more than 3.1 billion years ago. The evidence comes from geological signatures preserved in ancient samples. These findings refine models of early Earth dynamics. Further study of similar rock formations could clarify the timeline of interior water cycling. The work draws on preserved signatures to update understanding of planetary formation processes. Research combined high-resolution chromatography with human sensory panels to map how drying, storage, and cultivar choice alter cannabis volatile compounds. Different handling methods produce distinct shifts in perceived aroma profiles. The work details chemical changes that occurred during post-harvest processing and transport. Results clarify factors that influence final product sensory qualities. The mapping covers shifts tied directly to specific preparation steps. From organ system interactions in humans, we turn to coordinated behaviors at the microbial scale. Biologists at the University of California, San Diego identified an ejection mechanism in bacterial communities that allows groups to shed cells in response to threats. The process supports community persistence rather than relying on individual cell survival. This capability appears across soil, water, and microbiome contexts. The discovery provides a new angle on bacterial resilience mechanisms. Now, here's something most people get wrong about how bacteria handle stress. You've probably heard that bacteria are simple, passive cells that mostly drift or divide until external conditions kill them off. In reality, communities of bacteria actively jettison individual cells as a deliberate survival tactic when threats appear. Right now, microbial populations in soil, water, and even parts of the human microbiome are running versions of this ejection process to shed compromised members and preserve the group. One memorable detail is that this coordinated shedding happens at scales invisible to the naked eye, yet mirrors the logic of larger organisms abandoning damaged units. The practical takeaway is to remember that microbial resilience often comes from collective behaviors rather than individual toughness. Future reports on probiotic design or environmental microbiology will increasingly focus on these group-level strategies instead of single-strain properties. Researchers merged machine learning with quantum physics calculations to identify two new superconducting materials and speed screening of additional candidates. The method reduces the time required to evaluate potential compounds. Progress moves the field closer to identifying a material that superconducts at ambient temperature. Continued application could expand the catalog of viable candidates. The combined approach targets faster identification of room temperature candidates. A collaboration led by Professor Kotohiro Nomura at Tokyo Metropolitan University produced polyester amides from inedible biorenewable sources. The resulting films showed superior mechanical properties compared with common commodity plastics, while remaining chemically recyclable. The work demonstrates routes to high-performance materials from non-food biomass. Further scaling studies are implied by the reported synthesis approach. The films were evaluated for tensile strength against standard polyolefins. A newly identified mineral has been formally named spanoite in recognition of Tyler Spano's contributions to mineralogy. The mineral occurs in modest crystal sizes yet carries significance for understanding mineral formation processes. The naming follows standard conventions for honoring field advancements. Documentation of its properties adds to the catalog of known mineral phases. The formal description records occurrence details from the source location. While bacteria demonstrate collective decision-making, another study examines how similar functional needs produce varied physical solutions across insect lineages. Kobe University researchers examined how snatching forelimbs arose multiple times across insect lineages. Similar functional pressures produced varied structural solutions rather than convergence on one optimal form. The study introduces quantitative methods to track evolutionary dynamics of organs with comparable roles. This approach offers a framework for analyzing repeated trait evolution. Standard preparation of pseudo-pregnant recipient females often requires maintaining large colonies because only a fraction of mice enter estrus daily. A new method developed to bypass reliance on natural cycle detection lowers the number of animals held in stock. The approach also mitigates the Lee boot effect that suppresses cycles in group-housed females. Improved welfare outcomes accompany the reduction in colony size. The protocol targets efficiency in embryo transfer procedures. From evolutionary patterns in animals, we move to regenerative medicine questions involving cross-species tissue function. Multi-omics profiling of extracorporeal cross-circulation using gene-edited pig livers in 4 human descendants captured immune, coagulation, and metabolic responses. The data supply parameters relevant to future clinical use in liver failure support. This work advances regenerative medicine questions around tissue compatibility and function. Follow-up studies will monitor longer-term parameter stability. A bacterial enzyme isolated from marine sources was shown to support synthesis of protogenins, compounds with documented antibacterial, anti-cancer, and immunosuppressive activities. The discovery opens an enzymatic path to generating libraries of these natural product derivatives. Work focuses on expanding access to molecules already known for plant protection and therapeutic effects. Further enzyme characterization is underway. The enzyme enables new routes to bioactive compound production. After examining tissue-level compatibility, we look at how lifestyle factors combine or fail to combine in dementia risk reduction. Large-scale studies tracking diet, exercise, and social engagement reveal variable protective effects rather than uniform benefits. Some lifestyle factors demonstrate clearer associations with lower incidence than others. The findings temper expectations for single-factor solutions. Ongoing trials will clarify which combinations yield measurable cognitive outcomes. University of Tsukuba researchers used airborne LiDAR topographic data to quantify how soil thickness, landslide area, and slope gradient interact. Thicker soil layers correlated with larger failure zones under given slope conditions. The relationships improve hazard mapping accuracy. Continued high-resolution surveys could refine regional risk models. The topographic data link depth measurements directly to failure zone predictions. Kyushu University scientists synthesized a packaging nanomaterial from pumpkin peel waste that slows fruit deterioration and reduces mechanical damage during transport. The material was tested and reported in Food Research International. Waste stream sourcing lowers both cost and environmental footprint. Scale-up testing for commercial food applications is the logical next step. The nanomaterial extends produce shelf life through its derived structure. From human health patterns, we shift to a new computational tool for understanding proteins at scale. BetaDescribe, developed by teams at Technion and Tel Aviv University, translates amino acid sequences into natural language summaries of likely protein roles. The system was detailed in Proceedings of the National Academy of Sciences. It offers a route to annotate uncharacterized proteins at scale. Applications include accelerating functional genomics and materials design pipelines. From sequence to function mapping, we close with a broader look at how small mechanistic details accumulate across systems. The multi-omics dataset from gene-edited pig liver perfusion in human descendants supplies concrete immune and metabolic benchmarks for xenogeneic support strategies. These measurements directly address open questions in regenerative medicine around cross-species compatibility. The parameters will guide safety thresholds in planned clinical applications. What specific metabolic marker would you want tracked in the first living patient trials? Next time, we'll be watching for further details on how soil thickness measurements refine landslide hazard models. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Unintended consequences. True stories of good intentions that backfired. Find every show free at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.